we already have our first speaker who have joined our room, and let me talk about today's first topic and our first speaker, Dr. Anil Kumar Tiwary. He is today's first speaker who is going to deliver on the topic digestive physiology in chicken. Let me talk about first Dr. Tiwary. After veterinary graduation from Institute of Agriculture and Animal Science, Tiwary University, Nepal, Dr. Tiwary received Japanese government scholarship in animal physiology for studying Master of Science in Hiroshima University, Japan from 2003 to 2005. He is working as Assistant Professor of Veterinary Physiology at Faculty of Animal Science, Veterinary Science and Fisheries, Agriculture and Forestry University, Rampur, Chitwan, Nepal. Dr. Tiwary is also working as veterinary consultant at Brown Bird Hatchery, Bharatpur Forest, Chitwan since October 2000 AD till date. Professionally, he has experience of serving Nepal Veterinary Association Chitwan chapter as treasurer also. And as it is imperative to have a better understanding of digestive physiology in chicken before understanding the poultry nutrition and feed formulation, we have invited Dr. Tiwary to deliver his insightful talk on digestive physiology of chicken. Now I request Dr. Tiwary to proceed ahead with his insightful talk. Dr. Tiwary, now the floor is yours.
Hello. Hello. Hello.
Yes, Doctor, you are audible. You are audible.
Yeah. Thank you, Dr. Vivek, for giving me opportunity. Today, my topic is digestive physiology of chicken. Good morning, everybody. And I think we have only Nepalese participants or foreign participants also. Dr. Vivek, please. Hello.
Yes, foreign participants also.
Yeah. So
ideal to deliver in English only.
Okay. Okay. Thank you. Without wasting time, let's begin. Is it visible to everyone? Hello?
Yes, Raksha, it's visible.
Okay. Good working knowledge of the system and how it carries out its function is necessary for effective management of poultry flock, and therefore study of digestive system and the process of digestion and metabolism is important facet in the study of poultry husbandry. Mainly in the understanding of avian digestive system is essential to developing an effective and economical feeding program for your poultry flock. So before starting the main class of today's, that is about the feed formulation. So the basic thing is to understand poultry physiology, mainly digestive physiology. The digestive organ of domestic birds are obviously different in several respects from mammals. In general, the intestine of birds are relatively shorter than those of mammals. Presumably, such adaptations are important in reducing overall body weight in birds that fly. In adult chicken, the length of the entire digestive tract may be 200 centimeters or more. In poultry, teeth are absent and well-developed two-chambered stomach is present. The cecum is double and the rectum is short. linking the ileum with the cloaca, which is a common pathway for excretory and digestive wastage and the reproductive tract. These anatomical differences signifies the differences in digestive process. So let's see the digestive organs of poultry here. It starts from the beak and terminates to the cloaca. Here we have esophagus, throat, proventriculus, ventriculus or gizzard, duodenum, jejunum, ileum, and cecum, and very short rectum, and the last part is the cloaca. Now let's begin with the oropharynx. The oropharynx, that is mouth and pharynx, is combined cavity that extends from beak to the esophagus. The roof of this cavity is formed by palate, and there is only a hard palate which has long median cleft that connects with the nasal cavity. A shorter cleft, more caudally located, is the common opening for the auditory tubes. The floor of oropharynx is formed by mandible, tongue, and laryngeal mound, which is caudal to the base of the tongue. The laryngeal mound has a median slit. There is no epiglottis guiding the glottis. The triangular tongue moves a bolus within the oropharynx, propelling it into the esophagus when the bird swallows. Very little muscle tissue exists in the tongue. Its movements are produced by well-developed hyoid muscles. Taste buds are variably located in the oropharynx. Chicken may have as many as 300 taste buds. And in the chicken, salivary glands are present in many numbers and are well developed in chicken and turkey, but the secretion contains little amylase. Abundant saliva moistens ingested food and provides lubrication for boli when swallowed. Now, the next part is esophagus and the croup. Schematic of digestive tract beyond the oropharynx that begins with the esophagus is shown here. We can see this is the esophagus, and here it is dilated and forms the croup. The avian esophagus consists of a cervical and a thoracic region. It is wide and dilatable, thus serving to accommodate bulky, unmasticated food. Near the thoracic inlet, the cervical esophagus is a dilation that forms a pouch known as croup. And the function of croup is to store feed. When the proventriculus or gizzard is empty, the feeds bypass the croup. Little digestion takes place with the action of salivary amylase. Amylase activity at this site comes from either salivary secretion, intestinal reflux, or plant or bacterial sources. Starch is hydrolyzed within the croup, where it can either be absorbed, converted to alcohol, lactic acid, or other acids. Here we can see the proventriculus and the gizzard. In the thoracic region of the post-croup esophagus terminates in proventriculus. Mucus glands are abundant in the esophagus to provide lubrication for the food being swallowed. Sorry, I showed the figure before. The stomach, the proventriculus is the first of the two chambers that comprise avian stomach. The second chamber is the ventriculus, and it is also known as gizzard. The proventriculus is glandular stomach, and the ventriculus is the muscular stomach. Now let's see, the ventriculus is composed of opposing thin and thick muscle pairs, that is smooth muscles. A deep red color is due to a high concentration of myoglobin. The mucosal surface of ventriculus is lined with a thick cuticle called choline, a carbohydrate protein complex. This is the choline. Choline is formed when the mucosal secretion solidifies on the surface following exposure to the low pH in the ventriculus. The cuticle or choline protects ventriculus from the acid and proteolytic enzymes secreted by the proventriculus. The cuticle is continuously secreted at its base and is continuously eroded away on its surface. And this cuticle works like a sandpaper. The greenish or brownish color of the cuticle is due to the reflux of bile pigment from the duodenum. We can see grit in the ventriculus in most granivorous and herbivorous birds. It is used for grinding hard food between the thick muscles of ventriculus. Grit apparently is not essential for normal digestion, but digestion of hard food is slower and digestibility of a diet may be decreased without it. Normally, grit is ingested regularly, but if it is not available, food is retained for a longer period in the gizzard. Now comes the small intestine. The small intestine is continued caudally from the ventriculus by the duodenum. Division of small intestine into duodenum, jejunum, ileum are indistinct in case of poultry or chicken. There is duodenal loop as found in mammals. The vestige of the yolk sac, that is Meckel's diverticulum, is about midway in the small intestine and is used to indicate Meckel's diverticulum. It is used to indicate the division between jejunum and ileum. A well-defined network of blood capillaries, connective tissue, smooth muscle, and nerve fibers are present in lamina propria, but there are no lacteals, that is, blind beginning of lymph capillaries not present, which is present in case of mammals. The ileum ends with a circular ring of muscle tissue projecting into rectal lumen that appears to serve as a valve at the ileococolic junction. Here, this is the ileococolic junction. Entrance to the ceca are located immediately caudal to this ring. Rectum and cloaca. The rectum is relatively short and links the ileum with the coprodial compartment of the cloaca. The coprodium is the most cranial of the three cloacal compartments, followed in order by the urodium, and proctodium. Here in the figure, we can see these are the compartments. This one is coprodium, and this one is urodium, and this one is the proctodium. Here is the bursa fabricius, and this one is the opening of the vent. That is opening. This one is the vent. The three compartments are continuous. These compartments are continuous. Urinary and reproductive tracts empty into urodium, and proctodium opens externally through anus, that is vent. Bursa of fabricius, which is involved in immune function, projects dorsally from the proctodium. Now the accessory organs of digestion, liver and pancreas. Liver has two lobes, right and left lobes, and pancreas are accessory organs to avian digestion. Pancreas is located within the duodenal loop. There are three pancreatic ducts in domestic fowl that drain into the distal port of the duodenum. Gallbladders are present in chicken, turkey, ducks, and geese. And bile is transported to duodenum by two ducts, one from each liver lobe. The duct from the right lobe is the only one connected to the gallbladder. The bile duct drains into the distal duodenum near location of pancreatic ducts. Now, prehension and deglutition. In chicken, food is grasped by beak and brought into the mouth by repeated upward and downward movement of the head. Following prehension, swallowing is accomplished in three phases: oral, pharyngeal, and esophageal. In the oral phase, food bolus moves caudally into the pharynx by rapid rostrocaudal movement of the tongue. Bolus movement is assisted by caudally directed papillae. During the pharyngeal phase, the glottis and choanal and infundibular clefts are closed when the pharyngeal roof or the tongue are stimulated. The distance between oropharynx and esophagus is decreased by a forward movement of esophagus coupled with hyoid apparatus becoming concave and the tongue moving backward. Food particles are propelled from the tongue to the esophagus by further head raising and the tongue movement that are assisted by rostrocaudal movement of the laryngeal mound that contains rows of caudally directed cornified papillae. During esophageal phase, peristalsis within the esophagus moves the bolus towards the stomach. Swallowed boli enter the ventriculus when it is not filled. When the ventriculus is filled with food, esophageal muscle in the region of the crop relax and food enters the crop. Domestic fowl depends on gravity to assist in getting water into the pharynx for swallowing, because it cannot make its mouth for sucking the water. Motility of the gastroduodenal tract. In chicken and turkey, there is a rhythmic gastroduodenal contraction sequence that occurs at the rate of about 3 contractions per minute. A sequence includes the following in order of occurrence. The sequence is: first, contraction of thin ventricular muscle. Second, 2 or 3 peristaltic waves through the duodenum. And third one, contraction of thick ventricular muscle. And fourth, a peristaltic wave through the proventriculus. During each sequence, ingesta flows aborally into the duodenum by contraction of thin ventricular muscle and continues aborally by peristaltic wave of the duodenum. Ingesta flow orally into the proventriculus. Here, ingesta flows orally, that is in the opposite direction towards the mouth, into the proventriculus by contraction of thick ventricular muscle, which allows further mixing with proventricular secretions. This latter flow produces pressure changes in proventriculus that precedes its contraction, whereby ingesta are returned to the ventriculus. The next sequence begins with the contraction of thin ventricular muscle and flow of ingesta into the duodenum. Contraction of proventriculus and duodenum are dependent on intrinsic neural connection with the ventriculus. Extrinsic innervation does not appear to be involved in initiation of contraction or to be significant in the regulation of sequence. In addition to the above sequence of contraction that move contents orally and aborally, there is another oral movement of luminal content that occurs about 4 times per hour in turkeys. It involves a reflux of duodenal and upper jejunal content into the ventriculus. This activity permits mixing of intestinal content with gastric secretions. Ileal, colonic, and cecal motility. Peristalsis and segmenting contractions have been observed radiographically in the ileum. In the turkeys, these normally occurs at a mean frequency of about 4 per minute and 6 per minute in periods of more intense activity. The colon links the ileum with the coprodial compartment of cloaca. Antiperistalsis occur almost continuously in the colon. Function of antiperistaltic contractions are to move urine from cloaca into the colon and ceca for water reabsorption and filling of the ceca. There are major and minor contraction that occurs in the ceca. In the ceca, 2 types of contraction are there. The major contractions are associated with peristaltic contraction in the colon, whereby a series of major contraction is associated with cecal evacuation and a single major contraction is associated with defecation. The minor contractions perform a mixing function in the ceca. Cecal droppings have a chocolate brown color, homogeneous texture, and are distinguished from intestinal droppings that have a greenish granular textures appearance. 1 or 2 cecal droppings occur per day, while 25 to 50 intestinal droppings are formed. Here we can see the difference between intestinal dropping and cecal dropping. Here we can see the granular particles, but this is homogeneous mixture. And this type of feces is done only 2 times a day. The average daily production of feces from laying hen is about 100 to 150 grams. These fresh droppings are approximately 75% water and will air dry under favorable conditions to approximately 30% water. Secretion and digestion. Salivary, crop, and esophageal secretions. The salivary gland in chickens and turkeys are mucus secreting cells that primarily assist lubrication. Chickens secrete 7 to 30 ml of mucinous saliva per day. The mucosal surface of the esophagus contains glands that secrete mucus for lubrication of its surface. Mucus is also secreted by the crop in gallinaceous bird, but amylase is not present. A significant amount of starch digestion occurs in the crop as a result of bacterial action. Non-bacterial digestion of carbohydrates also occurs in the crop and results from amylase activity at the site that comes from intestinal reflux. A more thorough mechanical and chemical digestion occur in the stomach and intestine. Gastric secretion. Two types of glands are predominant in proventriculus. Simple mucosal gland that secrete mucus, and second one is compound submucosal gland, which are functionally homologous to the chief and parietal cell of the mammalian stomach that secretes mucus, hydrochloric acid, and pepsinogen. Pepsinogen is converted to pepsin in the acid environment, and pepsin produced begins the hydrolysis of protein molecule to polypeptide. The pH of gastric juice is 0.5 to 2.5, is appropriate for good peptic activity. The pH of gastric content is higher, that is 4.8, due to the presence of ingesta. The grinding action of the ventriculus not only reduces food particles size, but also mixes digestive fluid with the food. Grinding is facilitated by the presence of grit in the diet containing large particles. The presence of grit is not necessity when commercially prepared feeds are fed, because the feed grinding process adequately reduces the size of ingredients, thereby facilitating gizzard activity. Intestinal, pancreatic, and biliary secretions. The small intestine is the primary site of chemical digestion, accomplished by pancreatic, enzymatic, and microbial activity, as well as by intestinal secretions. The exocrine pancreas secretes lipase, amylase, and precursor of proteolytic enzymes, trypsinogen, chymotrypsinogen, A, B, and C, procarboxypeptidase A and B. The proteolytic enzymes are not activated until secreted into the intestinal lumen, because trypsin inhibitor is secreted by the pancreas to ensure proteolytic inactivation within the pancreas, thereby preventing autodigestion. Trypsinogen is activated within the intestinal lumen to trypsin by enterokinase and intestinal enzyme secretion. Enterokinase. Following action of trypsin activates other proteolytic enzyme precursors. Trypsin and chymotrypsin hydrolyzes specific bonds within large protein molecules and polypeptide to produce oligopeptides. In the intestinal lumen, carboxypeptidases release free amino acids from the oligopeptides. Aminopeptidase and dipeptidase are synthesized in the cytoplasm of enterocytes and perform cross-border hydrolysis of oligopeptides. These two enzymes are produced by enterocyte, aminopeptidase and dipeptidase. The primary carbohydrate in poultry feed is starch, of which there are two forms, amylose and amylopectin. Both forms are rapidly hydrolyzed by alpha amylase secreted by pancreas to form maltose, a disaccharide composed of two glucose units. The principal carbohydrate enzymes are maltase and isomaltase, and their substrates are maltose and dextrins, respectively, which generate glucose and sucrose. And the substrate used is sucrose, which generates glucose and fructose. Now the third one is fat. Fats are hydrolyzed to fatty acids and glycerol before absorption. This hydrolysis is accomplished by pancreatic lipase and by the action of bile salt that emulsifies fat and activates pancreatic lipase. In addition to digestive enzymes secreted by exocrine pancreas, an aqueous solution of bicarbonate is also secreted, which acts to neutralize the acidic gastric chain. The pH of avian intestinal tract increases from oral to aboral from about 5.6 to 7.2. A pH of 6 to 8 is considered optimal. In this figure, we can see the pH rises from oral to aboral end. Here we can see here in the colon, it is about 7. And here we can see the transit time. In the probe, it is about 50 minutes, and here the pH is 5.5. The proventriculus and gizzard, the transit time is 90 minutes, and pH is 2.5 to 3.5. In the duodenum, transit time is 5 to 8 minutes, and the pH is 5 to 6. Jejunum, 20 to 30 minutes, and pH is 6.5 to 7. In the ileum, it is 50 to 70 minutes, and pH is 7 to 7.5. In the colon, the transit time is 25 minutes, and the pH is 8. Secretion of bile into the duodenum also aids in neutralization of chyme. Bile salts are readily absorbed through the intestinal wall, allowing their recirculation and reuse. Factors affecting the secretions, that is, pancreatic secretion is controlled by nervous and hormonal components. The nervous components has a cephalic phase, whereby sight of food increases its secretion. This response is mediated by cholinergic fibers of the vagus nerve. Pancreatic juice refers to both aqueous and enzyme components. Pancreatic aqueous secretion is stimulated by vasoactive intestinal peptide in response to entry of acidic chyme into the duodenum. Avian vasoactive intestinal peptide is involved in secretory regulation, analogs to secretion in mammals. Release of cholecystokinin initiates the secretion of pancreatic enzymes. VIP does not stimulate pancreatic enzyme secretion. In addition, distension of the proventriculus with peptone, product of partial protein digestion, not only stimulates pancreatic enzyme secretion, but also secretion of secretion of aqueous component. This effect is mediated by gastrin releasing peptides. Diet can influence the secretion rate of pancreatic enzymes. If the content of carbohydrate and fat in the diet are increased, pancreatic amylase and lipase secretions are increased. Also, increasing protein content of the diet increases chymotrypsin activity in the duodenum and jejunum. Cecal function. Little or no digestion takes place in the large intestine, and only about 10% of most diets receive cecal digestion. The most noticeable digestive functions of the ceca are reabsorption of water from refluxed urine and microbial digestion of cellulose. Two important: reabsorption of water from refluxed urine and microbial digestion of cellulose. Ceca are filled by anti-peristaltic activity of the colon. We have already talked that in the colon there is anti-peristaltic movement which propels intestinal content into the ceca. A circular muscular ring of ileum projects into the colon, and its contraction, sphincter-like action, effectively prevents reflux of colonic material into the ileum. Urine is refluxed from the cloaca into the colon, and it enters the ceca via colonic antiperistalsis. The nitrogenous component of avian urine is uric acid, which is degraded for utilization of nitrogen by microflora, which are present in the ceca. Water reabsorption from the reflux urine is an important function of the ceca. Absorption. Most absorption of carbohydrate, amino acids, and fatty acids occur in the duodenum and proximal jejunum. Glucose absorption occurs predominantly in the duodenum and jejunum via passive transport, that is high concentration to low concentration. Active transport of glucose, that is carrier mediated sodium potassium ATPase dependent, occurs primarily in the ileum, where luminal glucose concentrations are lower than in the duodenum and jejunum. Significant active absorption of glucose also occurs in the proximal cecum. Amino acids and peptides are also absorbed from the duodenum and jejunum via sodium potassium ATPase energy dependent co-transport processes. Carbohydrates and proteins are rapidly digested, and glucose and amino acids can be analyzed in portal blood within 15 minutes after eating. Now, the fatty acids absorption occur in the distal half of the jejunum and to a lesser extent in the ileum, rather than duodenum and proximal jejunum as noted for carbohydrate and amino acids. This is due to the location of bile duct entrance near the distal duodenum. That is, fat cannot be absorbed before mixing with the secretion of liver, that is bile. Accordingly, emulsification of fats is delayed. Fatty acids enter the enterocytes and are re-esterified to triglycerides, given a protein covering that makes them water soluble and packaged into the protomicrons. In the mammals, we have chylomicrons. Here we have protomicrons. These are water soluble. Protomicrons facilitates transport of water insoluble triglycerides. In mammals, re-esterified triglycerides are packaged into chylomicrons, which enter the central lacteals of the villus for delivery to the blood. Birds do not have lacteals in their villi, so protomicrons are absorbed directly into the hepatic portal blood supply. Volatile fatty acids arise from microbial decomposition of uric acid in the ceca. Acetate is predominant, but some propionates and butyrate are also present. Volatile fatty acids are absorbed from the ileum and ceca by passive transport. Urine formed by kidneys is excreted into the urodium portion of the cloaca. Urine is refluxed from the urodium into the coprodium and then into the colon and ceca via antiperistalsis. Urine mixed with feces and water may be absorbed from the colon and ceca to maintain osmotic equilibrium of the extracellular fluid. Domestic fowl do have a sense of taste, but it is characterized by general indifference to categories human recognize, that is as sweet and bitter. It was mentioned earlier that taste buds are variably located in the oropharynx and that chickens may have as many as 300 taste buds. On an otherwise inadequate diet, birds are indifferent to sucrose solutions. However, if caloric intake in the feed is restricted, a chick will select a sucrose solution and increase fluid intake to make up the efficiency. This nutritional choice was not made when an isocaloric solution of fat or protein was offered. Domestic fowl depleted of protein avoid a solution of casein and select only water, apparently because of the taste. In other tests, feed that was rendered so distasteful that it was totally avoided in a choice situation did not influence intake when there was no alternative. However, response to taste quality was modified by hunger, whereby offensive had to be increased almost 10-fold in no choice situation to effect or reduced intake over an extended period. And now, thank you very much. Hello.
Thank you, Dr. Tiwary, for your wonderful presentation. Now, I would like to request you to go through chat box if there is any question.
Hello.
Okay. There is one question regarding your
Hello.
presentation.
Pardon?
Am I audible?
Yeah.
There is a question in chat box regarding your presentation.
Yeah.
Is feeding grit necessary in both broiler and layer? And how is grit intake ensured in case of pellet feed?
Is feeding grit necessary in both broiler and layers? How is grit intake ensured in case of pellet feed? Yeah, this one is for the broiler birds. Feeding grit is not so much necessary, but for the layers birds, grit feeding is necessary. We can see many commercial breeders. They produce commercial chicks and they recommend to feed grits of appropriate size to their grower birds so that it helps in the development of gizzard. And if gizzard is developed properly, and the birds can perform better while it comes in lay. So feeding grits to the layers is necessary, and also for the laying hens it is necessary because the release of calcium is slow in case of grit. But if we give the calcium in powdered form, it cannot be retained in the intestine for longer duration, and it is not available for the birds for making cells. So feeding grits for to the layers grower birds and laying birds is necessary.
Thank you very much for your answer. And there was only one question in chat box, and if regarding this presentation, digestive physiology of chicken, if anybody have any question in their mind, they can simply unmute their microphone and put question. Okay, there is again one more question. How is
digestive enzyme activity during feed restriction and molting? Yeah, in the digestive enzymes, the secretions decreases, because if there is no food, the enzyme is not secreted. That is, without a stimulus, there is no response of that particular gland. So the enzyme activity is restricted. It is not same as it is in the usual cases. Now, next question is, at what age in the case of commercial layers should we start feeding grit? We can feed commercial layers at what age? From the beginning, after after 3 weeks, we can start feeding grits. But the size of the grits should be according to the age. As the age increases, the size of the grits should also be increased.
Okay, thank you for your answers, Dr. Tiwary, and I hope all the questions are addressed now. And thank you very much for your wonderful, insightful talk on digestive physiology of chicken. Thank you very much for accepting our invitation as a speaker and for your delivery on this very important topic, because it is not possible to understand nutrition and feed formulation without having the basic understanding of the digestive physiology of chicken.